Welcome, everyone. Today we're going to be exploring the histology of the urinary and reproductive systems. These systems share important structural and endocrine connections, so we'll look closely at how their microscopic anatomy supports filtration, hormone production, gametogenesis, and reproduction. You'll encounter many epithelial variations, specialized cells, and structural arrangements, and today's lab will help you identify them microscopically. Looking at the urinary system first, the urinary system consists of the kidneys, ureters, bladder, and urethra. The kidneys do most of the functional work, filtering blood, balancing electrolytes, and regulating blood pressure, as well as producing urine. The remaining structures simply transport or store urine. Histologically, you'll see transitions from simple epithelia in the kidney to transitional epithelium in the ureters and bladder. The kidney contains millions of nephrons, which are the true functional units. The cortex of a nephron, the outer layer, houses renal corpuscles and convoluted tubules, while the medulla is organized into pyramids, which are made up of the loops of Henle, as we see here, and collecting ducts. We'll see how this organization creates a counter-current exchange system that allows for precise control of water and salt. The nephron is a long tubular system with distinct segments, each with a unique epithelial specialization. Histology will show how microvilli, mitochondria, and or cell junctions reflect the reabsorption or secretory roles of different segments. Filtration begins at the corpuscle. Then filtrate moves through the PCT, loop of Henle, DCT, and finally the collecting duct. The renal corpuscle contains, as you can see here in this image, the glomerulus and Bowman's capsule. Here, filtration occurs through fenestrated capillaries, a basement membrane, and a podocyte foot processes. Blood enters through the afferent arteriole, here, and exits through the efferent arteriole, here. Important for maintaining pressure. Under microscopy, look for the clear urinary space in the tuft of capillaries. Several specialized cells work together here. The fenestrated endothelial cells, podocytes forming filtration slits, simple squamous, partial epithelial cells that line Bowman's capsule, and mesangial cells that provide structure and phagocytic activity. Mesangial cells also modulate filtration and keep the basement membrane clean. And this is the capsule that you can see here, the capsular space in white, and then the glomerular capillaries. Podocytes are kind of iconic as they have these large cell bodies and interdigitating foot processes, as seen here. These foot processes can sometimes be called pedicles or pedicels. They create these slits, and these slit diaphragms form the final selective barrier for filtration. Damage to podocytes is a hallmark of nephrotic syndromes, which you'll recognize by effacement of these foot processes. The PCT reabsorbs the majority of the filtrate, like glucose, amino acids, electrolytes, and water. Histologically, these cells are large, eosinophilic, and contain abundant microvilli, which forms somewhat of a brush border. The lumen often appears fuzzy or collapsed, and their mitochondria and basal infolding support an extensive active transport system. The loop of Henle has both thick and thin segments in both the descending and ascending sections. Each are adapted for either active ion transport or passive diffusion. The loop itself establishes osmotic gradients, which are essential for concentrating urine. Thick segments resemble proximal or distal tubules, while thin segments are simple squamous epithelia. In the descending limb, the epithelium is permeable to water, allowing passive water reabsorption. The ascending limb is impermeable to water, but transports sodium chloride either passively or actively. The combination enables the counter-current multiplier mechanism. And the distal convoluted tubule is involved in fine-tuning electrolyte balance. It has much fewer microvilli, so the lumen appears cleaner. This is the major site for sodium reabsorption and potassium secretion under aldosterone control. You'll also see mitochondria and basal folds supporting active transport. This site kind of emphasizes the aldosterone-dependent activity of the DCT as the distal regions respond dynamically to hormonal cues to adjust sodium retention and potassium secretion, which is key in acid base and blood pressure regulation. The collecting duct shows clear cell borders, lighter cytoplasm, and visible lumens. They respond to antidiuretic hormone by inserting aquaporin channels for water reabsorption. Different cell types, principal cells, and intercalated cells manage water balance and acid-base regulation. As ducts descend towards the papilla, the epithelium transitions to a more columnar type. As these ducts drain then into the minor calyx, and this region is crucial in the final concentration of urine. This slide is helping just visualize the two major regions of the kidney, the cortex with renal corpuscles up top more externally and convoluted tubules versus the medulla with straight tubules and collecting ducts. Identifying these zones is foundational for kidney histology. Here you can see densely packed corpuscles and tubules. We see the corpuscles here with the glomerulus inside and then all these tubules surrounding. The presence of glomeruli is the easiest way to identify the cortex. Proximal tubules will dominate the field and appear larger as well as more eosinophilic. You'll see parallel arrays of loops of Henle and collecting ducts in the renal medulla. This is a longitudinal section. No corpuscles should be present, and differentiating thick versus thin limbs is key. Thin limbs are simple squamous, while thick limbs have cuboidal cells. As you can see, these are cuboidal cells here. This just continues to highlight the tubule parallelism. Watch for vascular components as well. The vasa recta, which runs alongside the loops. We can see this here with red blood cells inside, and that reinforces countercurrent exchange. A lobe includes a medullary pyramid here, plus its overlying cortical tissue. So this would be considered a lobe. Each pyramid drains at its papilla, this area here, and understanding the lobal anatomy helps to anchor 3D orientation and microscopic views. Transitional epithelium or uroepithelium is specialized for stretching. We've learned this when we learned epithelial cells. Our dome cells will flatten as the bladder fills. Uroplakins reinforce the apical membrane, which creates a barrier that's impermeable to water and ions. Recognizing a relaxed state here versus a stretched state here is key in histology. Our ureter has transitional epithelium and smooth muscle layers arranged in an unusual way, as there's an inner longitudinal and an outer circular, but with spiraling patterns. These allow for peristalsis to move urine towards the bladder in accompaniment with gravity. And you can see here in the urinary bladder, we have thicker, smooth muscle bundles, the detrusor muscle. We have a highly folded uroepithelium or urothelium where the bladder is empty. And as the bladder fills, the folds flatten and the dome cells stretch.